Dear students, in this session we are going to discuss some of the organisms which doesn't fall in the Whittaker's 5 kingdom classification. So, in Whittaker 5 kingdom classification, kingdom Monera contains all prokaryotic organisms, kingdom Protista contains all unicellular eukaryotic organisms, kingdom Fungi or kingdom Mycota which contains all heterotrophic multicellular organisms, except yeast. Yeast is unicellular. Other are multicellular. Now coming to the kingdom Plantae. So kingdom Plantae contains all photosynthetic organisms. Kingdom Animalia contains all heterotrophic multicellular organisms. So here viruses. Viruses doesn't fall under these categories because the characteristics of these viruses is quite different from those organisms. So viruses are acellular obligate parasites having protein coat and genetic material. So when you are talking about the virus cannot replicate on their own like that of the other organisms because the virus doesn't contain the cell structure. So due to the lack of the cell structure, the virus cannot replicate on their own. So for their replication, it requires host. The host may be either plant or animal or bacteria or human. So normally other organisms can reproduce on their own because they have the cell structure. So the metabolic activities will be taking place inside the cell and the reproduction takes place. So here the viruses cannot replicate on their own. They depend on host. The host may be plant, animal, human or bacteria. So the viruses which infect plants are called plant viruses. The viruses which infect animals are called animal viruses. The viruses which infect algae is called phycophages. The viruses which infect fungi is called mycophages. The viruses which infect bacteria are called bacteriophages. So there are different types of viruses. So the viruses contain either DNA or RNA as the genetic material, but not both. So when you are talking about the virus is both living and non-living. Why? So whenever the virus infects the host cell, then only it will have the life. When the virus cannot infect the host, it doesn't have the life. So here you should remember one thing. So the virus is both living and non-living. Why? So whenever the virus is outside the host cell, it doesn't have the life. It is like a dust particle or it is like a non-living thing. So once the virus can enter into the host, it will use the host mechanism and it will replicate. So they are acellular. They do not contain the cells. Hence they are called acellular. They are obligate parasites. So they depend on other organisms for their survival. So they are obligate. Obligate means strict. They are very strict parasites. They depend on living organisms for their growth and development. Next, they have the protein coat and genetic material. So when you are talking about the virus has only two things, that is protein and genetic material. It does not have the metabolic activities. It does not have the cell. It is not having any enzymes. It is not having any cell organelles. That you should remember. So it is having only two things. One is the genetic material. Another one is the protein coat. So virus is having protein coat and genetic material. So what is genetic material? The genetic material may be either DNA or RNA. DNA means deoxyribonucleic acid. RNA means ribonucleic acid. So the genetic material is either DNA or RNA, but not both. So if you are talking about any virus, the virus contains either DNA or RNA, but not both. Either it is having the double stranded DNA or single stranded DNA, or single stranded RNA or double stranded RNA, but not both. So the viruses having either DNA or RNA. The viruses which are having DNA as the genetic material, they are called as DNA viruses. The viruses which contain RNA as the genetic material, they are called RNA viruses. So here the genetic material is either DNA or RNA, but not both, which is infectious. So whenever the virus can enter into the host, it will cause the infections. How it can cause the infections? The virus is having only two things, that is protein coat and genetic material. Here the protein coat is not infectious. Genetic material is infectious. Protein coat is not infectious. So whenever the virus infects the host, the genetic material will enter into the host and causes infection. The protein coat does not enter into the host. Whenever the virus infects the host, only the genetic material will enter into the host. The protein coat or the capsid, that does not enter into the host. Next, they require host for their survival. I already said they cannot replicate on their own. So they require host. The host may be either plant or animal or bacteria or human. You know that so many viruses are there which can cause infections. So here cold, if you take the common cold, that is mainly caused due to the virus only. So here they require host for their survival. Without host they cannot survive. Next, virus means venom or poisonous fluid given by Louis Pasteur. Louis Pasteur is the scientist who has given that name venom. Virus means venom or poisonous fluid, that is nothing but genetic material along with protein coat. They are the nucleoproteins. Viruses are nucleoproteins simply. Nucleo means they contain the nucleic acid. Protein means they contain the protein. What is a protein? A chain of amino acids. A polymer of amino acids is called protein. So Ivanovsky recognized TMV. TMV means tobacco mosaic virus. So TMV means tobacco mosaic virus. So Ivanovsky recognized the TMV, tobacco mosaic virus. Next, MW Bezernik demonstrated that extract of infected plants cause infection in healthy plants and called the fluid as contagium vivum fluidum. So Bezernik demonstrated that extraction of infected plants. Here, once the virus can infect the tobacco plants, this tobacco mosaic virus infects the tobacco plant, what happens? The genetic material will enter into the tobacco plant. Here the tobacco plant can act as host for the virus. So that fluid, whenever this infected plants can infect the healthy plants, that fluid that enters into the host, that fluid is nothing but contagium vivum fluidum. W.M. Stanley showed that viruses could be crystallized and contain proteins and they are outside the specific host. Here once again, Bezernik demonstrated that extract of infected plants. What is infected plant? If the virus that infects the host plant, that is called infected plant. So the infected plants cause infection in the healthy plant. Whenever the infected plants come in contact with the healthy plant also gets infected. How? The genetic material present in the virus can enter into from the infected plant to the host plant, which means the virus will enter from the infected plant to the healthy plant, and the healthy plant becomes infected. And that fluid, the fluid that is entering into the healthy plant from the infected plant is called contagium vivum fluidum. Next, W.M. Stanley showed that viruses could be crystallized and contain proteins and they are outside the specific host cell. Here the viruses can be crystallized, they can be in the form of crystals. So here they contain proteins, that crystal contains the protein, they are outside the specific host. I already said whenever the virus infects the host cell, the genetic material can enter into the host, protein does not enter into the host cell. So they are outside the specific host, the proteins become outside, the genetic material will enter into the host. Next, plant viruses have single-stranded RNA. So here, normally the plant viruses have single-stranded RNA. So animal viruses have either single-stranded RNA or double-stranded RNA, or single-stranded DNA or double-stranded DNA. So single-stranded RNA means it is single-stranded. Double-stranded means which contains two strands like this, genetic material. We are going to discuss this DNA and RNA in detail later. So plant viruses have single-stranded RNA normally. Animal viruses have single-stranded RNA or double-stranded RNA, single-stranded DNA or double-stranded DNA. Bacterial viruses have usually double-stranded DNA, rarely RNA. Normally bacterial viruses. What are bacterial viruses? The viruses which infect bacteria are called bacterial viruses. Bacterial viruses are also called bacteriophages. What are bacteriophages? Very important. The viruses which infect bacteria are called bacteriophages. So viruses are very, very small when compared to the bacteria. Bacteria is bigger than the viruses. Viruses are small, bacteria is bigger. So virus can infect the bacteria. Are called bacteriophages. Next, a protein coat called capsid made up of small subunits called capsomeres protects the nucleic acid. When you are talking about the viruses are nothing but nucleoproteins, we already discussed. So the protein coat, so if you are talking about the virus, it contains the nucleic acid and the protein coat, only two things. The nucleic acid is nothing but DNA or RNA and the protein coat. Here we are talking about the protein coat. So protein coat called capsid. So protein coat is also called capsid in the virus. So what is a capsid? The protein coat present in the virus is called capsid. Capsid is made up of small subunits called capsomeres. If you see here, this is the TMV structure, tobacco mosaic virus. So it is having the single stranded RNA. If you see this structure, this is the single stranded RNA. And here the protein coat called capsid. This is called capsid. This structure is called capsid. From here to here, this complete structure is called capsid. That is the protein coat. This structure, this spiral structure is called genetic material, RNA. I already said the virus contains only two things, genetic material, that is RNA here, and the capsid, that is nothing but protein. The capsid is made up of capsomeres. What is a capsomere? Here already said, capsid is made up of small subunits. This is the small subunits. Each small subunit is called capsomere. So this capsomere protects the genetic material. This is the capsomeres. So capsid contains capsomeres. The capsomeres protects the genetic material. That is nucleic acid here. Capsomeres are arranged in helical or polyhedral geometric forms. So these capsomeres are arranged in two forms, either helical or polyhedral. Helical means hollow cylindrical like protein structure is called helical here. The hollow cylindrical protein structure which protects the genetic material is called helical. Hollow like structure, hollow protein structure is called helical. Polyhedral means which is having many heads, like polyhedral, many sides. Many sides of the protein can protect the genetic material. That we are going to discuss in the bacteriophase. T2 bacteriophase. Next, coming to here, TMV, tobacco mosaic virus. This is the virus which infects the tobacco plant. So here it is having only two things. I already said, that is the genetic material and the protein coat. The protein coat is called capsid. Capsid is made up of number of subunits called capsomeres. So in TMV, how many capsomeres are present? In TMV, 2130 capsomeres are there. This is one capsomere, 2, 3 like that. Okay, so 2130 capsomeres are present in the capsid of the TMV. Next, what is the molecular weight of each TMV particle, each virus particle? 39.4 into 10 power 6 daltons. This is the molecular weight of the TMV. So single stranded RNA, I already said, the genetic material is single stranded RNA. So when you are talking about the DNA or RNA, it is made up of nucleotides. Okay, number of nucleotides can combine together to form either DNA or RNA. So how many nucleotides are present here? 6400 nucleotides are present in the genetic material, that is RNA. In RNA, how many nucleotides are there? 6400 nucleotides are there. What is the molecular weight of the TMV? 39.4 into 10 power 6 daltons. How many capsomeres are present in the TMV capsid? 2130 capsomeres are present in the capsid. So each capsomere has 158 amino acids. So I already said this capsomeres are nothing but protein. Protein is nothing but chain of amino acids. Each capsomere contains how many amino acids? 158 amino acids are present in each capsomere. So it is a rod-shaped helical virus. It is a rod-shaped helical virus. I already said helical is nothing but hollow structure which protects the nucleic acid. Hollow protein structure which protects the nucleic acid. So what is the length of this virus? That is 3000 angstroms or 300 nanometers. 3000 angstroms or 300 nanometers. That is the length of the virus. Here diameter is 18 nanometers or 180 angstroms. So this is about TMV. Next, coming to the bacteriophase. So what is a bacteriophase? The viruses which infect bacteria are called bacteriophages or bacterial viruses. So the viruses which infect bacteria are called bacteriophages. So here it is a tadpole like structure with polyhedral head connected to helical tail. When you are talking about the bacteriophase, which type of bacteriophase it is? T4 bacteriophase. So it is a T4, name of the bacteriophase here it is T4 bacteriophase. It is a tadpole like structure. You know that it is a larva of amphibian. It looks like a larva of amphibian, that is tadpole like structure with polyhedral head connected with helical tail. I already said polyhedral means it contains many sides. The genetic material is present here and it is the protein capsid contains many sides, 1, 2, 3, 4, 5, 6 like that, many sides. So polyhedral head connected to the helical tail. So this bacteriophage contains mainly two structures, that is head and tail, which is connected with the help of collar. Head contains nucleic acid surrounded by protein coat. This is the head which contains nucleic acid surrounded by the protein coat. What is the nucleic acid here? DNA. DNA is the nucleic acid or the genetic material in T4 bacteriophage. So the head and tail. Head contains genetic material which is surrounded by the protein coat. Tail. Now coming to the tail. Tail is proteinaceous tube-like structure surrounded by sheath. This is called tail. This structure is called tail. It is surrounded by sheath. It is like a tube-like structure. And here the tail mainly consists of two ends. This is one end and this is the other end. At one end, tube is joined to the head. This is the head. This is the tail. Tail and head can be joined with the help of neck or collar. At one end, the tube is joined to the head by collar. So this is called collar. You see this structure, this is called collar. So head and tail can be connected with the help of collar at one end. The other end, it contains the base plate. It has the base plate with 6 tail fibers. Here these are called tail fibers. 6 tail fibers which help in attachment to the host. So this is about bacteriophage. So the bacteriophage, the best example here is the T4 bacteriophage. Here it contains, it looks like a tadpole like structure with polyhedral head connected to the helical tail. Head connected to the helical tail. Head contains nucleic acid. This is head which contains nucleic acid. What is the nucleic acid here? DNA. In case of TMV, the nucleic acid is RNA. In case of bacteriophage, the nucleic acid is DNA. So head contains nucleic acid, that is DNA, surrounded by the protein coat. Next, tail is proteinaceous tube-like structure. This is called tail. It is a proteinaceous tube-like structure surrounded by the sheath. Here it is surrounded by sheath. At one end of the tail, it connects to the head with the help of collar. At the other end, it contains the base plate. Again, it contains 6 tail fibers. These are called tail fibers. Tail fibers helps for the attachment of this bacteriophage to the host. That is bacteria. What is the host here? Bacteria is the host. Why? The viruses which infect bacteria are called bacteriophage. Here host is the bacteria. The virus which infects the host. So this is about T4 bacteriophage. Now coming to the reproduction in viruses. How the virus particles can reproduce? Because they are not having the cells, then how they can reproduce? For their reproduction, they require definitely host. For their survival itself, they require the host. Even for the reproduction also, they require the host. Without host, they cannot reproduce. How the viruses can replicate? Here, the viruses can replicate by 2 mechanisms. If the viruses, that is lytic cycle and lysogenic cycle. The viruses which replicate by lytic cycle or through the lytic cycle is called virulent phase. Are called virulent phases. The viruses which replicate through the lytic cycle are called virulent phases. The viruses which replicate through the lytic and lysogenic cycle are called temperate phases. So the viruses, virulent phase and temperate phases. Virulent. So replicate by viruses, replicate by lytic cycle are called virulent phases. Now coming to the temperate phases. Viruses replicate by or through lytic and lysogenic cycle are called temperate phases. So the viruses here, 2 types, virulent phases and temperate phases. The viruses replicate through the lytic cycle or by the lytic cycle are called virulent phases. The viruses which replicate through the lytic and lysogenic cycle are called temperate phases. Now, what is lytic cycle and what is lysogenic cycle? In case of lytic cycle, here, for example, this is the bacteria, this is the virus. Here the genetic material is present. So you know that this is the host, this is the virus. In case of lytic phase, what happens? Here the genetic material will enter into the host like this. The bacteria is having already genetic material. Now this virus genetic material will enter into the host, and this undergoes multiplication. This genetic material undergoes multiplication and new viral particles are formed within the host, and here the lysis takes place. Lysis means breakdown. Once the new viral particles are formed in the bacterial cell undergoes breakdown. So bacteria cell undergoes death. Next, what happens? All these newly formed virus particles will be released. That it takes place in case of lytic cycle. In case of lysogenic cycle, what happens? Here the bacterial cell does not undergoes lysis immediately. Here the genetic material will enter into the bacterial cell and incorporates with the genetic material of the bacteria like this. In case of lysogenic cycle, whenever the bacteria undergoes multiplication, the genetic material undergoes division, right? So here what happens? Here the genetic material undergoes replication. So whenever the bacteria undergoes multiplication, the genetic material undergoes replication, and one bacteria becomes 2 here, the genetic material also 2 here. Along with the bacterial genetic material, the viral genetic material undergoes multiplies like this. That takes place in case of lysogenic cycle. So in lytic cycle, what happens? The genetic material of the virus will enter into the bacteria, and the genetic material undergoes replication by using the host machinery. Host cell machinery, and number of virus particles are formed within the bacteria, and the bacteria cell undergoes lysis, nothing but breakdown. The bacterial cell undergoes breakdown and all the virus particles will come out. That takes place in case of lytic cycle. In case of lysogenic cycle, what happens? Once the viral DNA can enter into the host, it will integrated with the bacterial chromosome. In lytic cycle, the viral DNA does not integrated with the mixed with the bacterial chromosome. But in case of lysogenic cycle, the viral DNA integrated with the bacterial chromosome. That is the difference. Another difference, in case of lytic cycle, the host DNA undergoes breakdown. Bacterial DNA undergoes breakdown in case of lytic cycle. Once the viral DNA can enter into the host. In case of lysogenic cycle, what happens? The host DNA does not undergoes breakdown. Here what happens? The genetic material of the virus will be integrated with the host DNA. And whenever the host cell undergoes multiplication, here the viral DNA undergoes replicate along with the bacterial DNA. Later virus particles will come out. So this is how the reproduction takes place in the viruses. So this is about viruses. Next, coming to the viroids and prions. Viroids and prions also, they do not have the cell structure. So first coming to the viroids. So T.O. Diner discovered these infectious agents smaller than viruses. So who discovered the viroids? T.O. Diner. So T.O. Diner discovered the viroids. These are the infectious agents. They are smaller than viruses. So viruses are very, very small. When compared to the viruses, viroids are little bit small. So they contain only genetic material, that is RNA but not protein code. So they contain the naked RNA but not protein code. Normally in case of viruses, what happens? The protein code is surrounded by the protein code surrounds the genetic material. So the genetic material is surrounded by the protein coat in case of viruses. So the viruses are nucleoproteins. They contain genetic material and the protein coat, but viroids contain only the genetic material, that is RNA. It does not contain the protein coat. So viroids are the infectious agents that contain only genetic material, that too RNA, but not protein coat. Next, molecular weight is very, very low. The viroids molecular weight is low when compared to the viruses. So viroids cause diseases. Potato spindle tuber disease and chrysanthemum stunt disease are the examples. The diseases caused by the viroids are potato spindle tuber disease and chrysanthemum stunt disease. So in viroids, here are the 4 points here. T.O. Diner discovered the viroids. These are the infectious agents. So they are very small when compared to the viruses. The molecular weight is low. They contain only the genetic material but not the protein coat. The molecular weight is low and they causes diseases, that is potato spindle tuber disease and the chrysanthemum stunted disease. So this is about viroids. Now coming to the prions. So prions are the infectious agents, proteinaceous infectious agents which does not contain the genetic material. They contain only protein. That is the difference between the viroids and prions. In case of viroids, only genetic material is present, protein coat is absent. Here only protein capsid is present, genetic material is absent. Protein coat is present. So they contain only protein but not nucleic acid. Next, these are the infectious agents contain abnormally folded proteins which cause neurological diseases. The proteins are abnormally folded. So folding of proteins that we are going to discuss in molecular biology. So here infectious agents. So prions are the infectious agents which contain abnormally folded proteins cause neurological diseases. So some of the examples which causes prions causes diseases. Some of the examples of these diseases are scrappy disease of sheep, mad cow disease in cattle, Kuru disease. Kuru disease is also called laughing death disease. So this is about prions. So in prions are nothing but these are the proteinaceous agents which does not contain the genetic material. Here the proteins are abnormally folded and causes neurological disorders. So here the diseases which are caused by the prions are scrappy disease of sheep, mad cow disease in cattle, Kuru disease, that is laughing death disease. So this is about prions. With that we completed biological classification. Lichens we already completed. So this is about the topics in subtopics in the biological classification. In the next session we are going to discuss anatomy of the flowering plants.